Good morning, everyone. We have about 140 participants, so I think we will go ahead and start because I don't want our guest speaker to spend too much time waiting because he's joining all the way from the other side of the world, and it's quite late for him right now. And I kind of apologize if it is inconvenient for us to schedule this quite early morning. And again, we are battling about 13-hour time difference between our guest speaker who's joining us from United States. So in order to facilitate both parties, we decided to go ahead with 8 a.m. in the morning for Sri Lankan time. So welcome everyone for the third virtual webinar organized by the Nutrition Society as a part of our ongoing virtual webinar series. And today, with great pride and pleasure, I would like to introduce you to today's guest speaker, Dr. Dhananjaya, who is one of my dear friends. We have been friends since 2005 because we have attended the same university and faculty as undergraduate students. And even though we haven't seen each other for over about a decade, I believe we maintained our friendship throughout. And I'm very grateful when he agreed to deliver this talk to our students because he's going to talk about omega-3 fatty acids and its therapeutic potential in treating inflammation and autoimmune diseases, a very timely and a very interesting topic. And I believe our nutrition students will benefit greatly from his intellect, his experience and expertise in this area. So Dr. Dhananjaya, as I said, attended Faculty of Agriculture, University of Peradeniya for his bachelor's degree. And afterwards, he completed his master's in PGIA in the field of food and nutrition. And he left the country to attend the doctorate program at Wayne State University in United States some time ago. And ever since, he's been working as a postdoctoral fellow in different universities. And last year, he was working with this particular research area as a postdoctoral fellow in Michigan State University, one of the very prestigious universities in the United States, and where he received several accolades for his research with respect to omega-3. I'm sure he will talk about it in his slides, or he will probably bring forth his experience in terms of doing research in this area. And from this year, he's been working as a scientist in the bioscience immunology section in AstraZeneca USA as a major scientist, and continuing his work with respect to omega-3 in the field of unraveling its effects on autoimmunity and inflammation. So I don't want to spend too much time exchanging pleasantries because I'm sure Dr. Dhananjaya has a lot to talk to you. So I am going to invite him to deliver his talk today. And again, I would like to take this opportunity to thank him for graciously agreeing to talk to our students. I know because he's quite busy doing his research work and all the other work that is going around with him probably this time around. So we are very grateful that he agrees to give us a talk today. And again, I have to remind that it's quite late from his timeline. So again, thank you very much for agreeing to help us out and just to share your intellect and experience and expertise with our students. Dr. Dhananjaya, the screen is all yours. He has already shared his slides, so he will continue with his slides. And if you have any questions, I think we can take them at the end, or maybe you can type in the chat box. Ms. Parami and Ms. Senari, representing the Nutrition Society, will take questions and will continue the discussion at the end of the session. So that's all from me. Dhananjaya, whenever you are ready, the screen is yours. Thank you.
Thank you, Dushanti, for your introduction. And I'm very honored and excited to be here to share some of my latest findings on inflammation, autoimmunity, and omega-3 fatty acids. And also, I would like to thank organizers for inviting me this seminar. Especially, I want to thank to Shanti. We used to be in the same university as she mentioned. Uh, Let me go into my title. Today, my title is Unraveling Effect of Omega-3 Fatty Acid on Inflammation and Autoimmunity. Since audience is undergraduate students, I'm going to discuss this topic in very simple manner. If you have any questions or any doubt, you can ask me at the end of the presentation. You can use any language, Sinhala or English. I'm comfortable with both languages. This is my outline. First, I'm going to give brief introduction about inflammation. Then I'm going to talk about how unresolved inflammation drives toward the autoimmunity. Then I will compare and contrast effect of saturated fatty acid, omega-6 and omega-3 fatty acid on inflammation. Finally, I will discuss a case study related to lupus and fish oil. Let me give a brief introduction about inflammation. Inflammation is a body process of fighting against infection. Infection may be virus, bacteria, or fungus, and also injuries and toxin. Toxins could be environmental toxins such as silica, agrochemicals, and so on. And also it could be food toxins like food allergen and so on. During the inflammation process, it releases chemicals that triggers response from immune system. It is completely normal. It is a protective action and it's part of our host defense mechanism. Inflammation or inflammatory response is a cellular response. It involves activation of cells in response to presence of triggers. Most often we think infectious triggers like microbial component, but it can also be triggered by tissue damage, metabolic stress, and toxin. Especially, as I mentioned earlier, toxin could be environmental toxin, food toxins. And the link between triggers and cellular response is controlled by cellular signaling pathway. Receptors on the cells receptors on the cells recognize these triggers, which activate the intercellular signaling pathways and inflammatory genes. And genes produce different proteins and enzymes. Sometimes these protein directly have response on inflammatory response, and sometimes enzymes degrade some substrate and produce some lipid mediators and also some chemical mediators. This is what we measure as inflammatory response. Some of the inflammatory markers include TNF alpha, IL-6, IL-1, MCP-1, some different chemokines, inflammatory lipid mediators. Since I just spoke about inflammatory response markers, there are some drug targets that target on these inflammatory markers. If we can inhibit this inflammatory marker, it will help to reduce inflammation. This is just for information. You don't need to memorize these drugs. Let me explain inflammation process step by step. This is an inflamed finger. So what happened here is some damage to the surface of the skin, possibly with bacteria. Damaged area of the skin release some signals. These attract white blood cells from bloodstream to site of the damage. That's why you get swelling. That's why you get redness. That's why you feel heat. There are a lot of cellular activation at that site. Basically, body is trying to clearing up any microbes and activate the tissue repairing process. In this process, macrophages and neutrophils plays a key role. These are the 5 cardinal signs of inflammation that include heat, redness, swelling, pain, losses of function. Basically, you can see all these outcome in inflamed figure. Inflamed finger. So these are the finger. You can see redness, heat, swelling, pain, and losses of function. The cause root of these outcome is cellular response that I explained very first slide. Do you remember them? For recap, cellular response include recognized triggered by receptors on the cells activate a cellular signaling pathways in cell, activate inflammatory genes, and release of several chemokines and cytokines, which we measure as the inflammatory response. All together gives these outcomes, including heat, redness, swelling, pain, and losses of function. Let's see how inflammation makes changes over the time. Basically, trigger launch the initiation for inflammation. Can you remember the triggers? Microbes, toxin, tissue damage, and also oxidative stress. We can measure this intensity from inflammatory markers. I think in the very first slide, I spoke about these inflammatory markers. Sometimes this inflammation reaches to its peak and goes away within a minute. Sometimes it can take days, but it's self-limiting. This is known as resolution phase. I will come back towards the end of my talks. Usually, a self-limiting resolution is protective, it is normal, and it's part of host defense mechanism. In some scenarios, you will get strong triggers like strong virulent bacteria or like sometimes COVID virulent virus. So then you will have a strong inflammatory response. And then you have to take drugs to get relief. In that case, you have to take some anti-inflammatory drugs or some vaccine to cool down the inflammatory response. Sometimes there's no resolution. The high inflammation continues over the time. This is the danger type of inflammation. This is also known as silent inflammation, where inflammation is uncontrolled, excessive, continues without resolution. You can see tissue damage and other pathologies, and also it can lead to autoimmunity. A good example of this classical inflammatory condition is rheumatoid arthritis, when you have unresolved inflammation in joints, which ultimately cause for damage to bones. Here is the end results. You can see swelling, losses of function, pain, and so on. This is the another great example. Inflammatory bowel disease. Leaks in gut epithelial could lead to unresolved inflammation in endothelial tissues of digestive tract. This is the end results. You can see here swelling, losses of function, pain, and redness, and so on. Acute inflammation without resolution also can be fatal. For example, when you expose to virulent bacteria infection without resolution, and also one of the best example is COVID vaccine, and you will get strong inflammation. In that case, definitely you have to get the drugs. If not, if it continue, definitely it is lethal. So this is the one of the best example just published in the Nature Scientific report. And here, patient with bacteria infections in intensive care were tracked for their blood inflammatory markers over 28 days. 49 of these individuals survived, and unfortunately, 17 died. So what they found was inflammatory markers were high in non-survived groups compared to survivors. In this case, there's no resolution. So what I want to emphasize here is whereas the inflammation is uncontrolled or excessive, continue without resolution, we get tissue damage and develop autoimmunity. Now I believe you can understand the relationship between inflammation and autoimmunity. Here are the common examples for chronic inflammation-related disease: atherosclerosis, obesity, chronic COPD, uh, autoimmune diseases, Alzheimer, arthritis. There are several diseases related to chronic inflammation. So I have already completed section 1 and section 2. Now I'm going to compare and contrast the effect of saturated fatty acid, omega-6 fatty acid, and omega-3 fatty acid on inflammation. Fatty acid can be classified into major two groups: saturated fatty acid unsaturated fatty acid. Unsaturated fatty acid can be further classified into major two groups: omega-6 fatty acids and omega-3 fatty acid. The main difference between saturated fatty acid and unsaturated fatty acid is, like, saturated fatty acid does not have any double bond, but unsaturated fatty acid have several double bonds. They can be one unsaturation, then it's known as monounsaturated fatty acid. If there's several unsaturated fatty acids, then they are known as polyunsaturated fatty acids. Effect of saturated fatty acid. Let's talk about the effect of saturated fatty acids on inflammatory markers. So this is a recapping. Here onward, I will mention several markers for inflammatory response that I mentioned in introduction slides, that include TNF alpha, IL-6, IL-1, some chemokine, and lipid mediators. They are just indicators for inflammatory level in the cells, animals, and human. So you just need to get idea. You don't need to memorize these names. So again, I want to emphasize, I will use these inflammatory markers: IL-6, TNF alpha, COX-2, IL-1 alpha, IL-1 beta, PGE2. High inflammation means high expression of these markers. Low inflammation means low expression of these markers. Now I'm going to focus uh, with how a saturated fatty acid affect on inflammation. Here I choose one of the classical initial study that was done in 2004. Later, several researchers validate this finding using a series of experiment. But since I want to give the credit for initial researchers, I choose this paper. Basically, they measured the effect of saturated fatty acid on inflammation. So what they did was, like, they incubated human coronary endothelial cells with palmitic acid, then measured the production of different inflammatory markers, including IL-6. So basically, palmitic acid is saturated fatty acid, and again here we measure the IL-6, is the indicator for inflammation. What they found was palmitic acid induced IL-6 expression in human coronary artery cells. There are two types of cells they choose: endothelial cells and smooth muscle cells. So let's look at the IL-6, which is the black bar. You can see very high level of IL-6 production in endothelial cells as well as smooth muscle cells after incubation with palmitic acid. But in the control group, they don't see that much IL-6 expression. Then they want to measure the effect of palmitic acid on dose effect of palmitic acid on coronary artery endothelial cells. What they found is palmitic acid dose dependently induced IL-6 expression in human coronary artery endothelial cells. Let's look at the 500 micromolar concentration. You can see very high level of IL-6 production in both endothelial cells and smooth muscle cells. Here, palmitic acid, again, palmitic acid is saturated fatty acid, and IL-6 is indicator for inflammation. So let's move into another classical paper with in vivo studies that was published in 2001. This paper is the first time they identify the inflammatory mechanism of saturated fatty acid. These two papers have done great impact on advancement of lipid research since last 20 years. Basically, what they did was they incubated murine cells with different saturated fatty acids and then measured the production of different inflammatory markers, namely COX-2. COX-2 again is the indicator for inflammation. They used the different saturated fatty acids. 12 mean lauric acid. This 14 means myristic acid, palmitic acid, stearic acid. And you can see here what they found was lauric acid induced maximum COX-2 expression. So then they observed dose effect of lauric acid on these cell type. And they found when they increase the lauric acid concentration, COX-2 expression is increasing. Let's move into some clinical studies results. A limited number of observational studies have investigated the relationship between saturated fatty acids and circulating inflammatory markers. They found serum fatty acids were positively correlated associated with IL-6 concentration in overweight individual. You need to memorize this is in overweight individual. When saturated fatty acid content is increasing in overweight individual, their IL-6 content also increase. Interestingly, they did not see any relationship between saturated fatty acids and IL-6 in lean individual. That means physical activity is very important in eliminating inflammatory effect of saturated fatty acids. For summary, I have already talked about an inflammatory nature of saturated fatty acids on cells, animal, and this is human studies. Let's talk about unsaturated fatty acids. There are main two types of unsaturated fatty acid, omega-6 and omega-3 fatty acid. First, I will talk omega-6, then I will move into omega-3 fatty acid. There are main two types of omega-6 fatty acid, linoleic acid and arachidonic acid. When we take our food, with these fatty acid, they go through the digestive tract and they digest those fatty acid and transfer into the blood. Through the circulation, these fatty acid can transfer into the cell membrane. When cells get external stimuli, it could be, can you remember the triggers? It could be microbes, it could be toxins, and also oxidative stress or tissue damage. Then what happened? These arachidonic acid in cell membrane further metabolite into small lipid mediators. They include prostaglandin, thromboxane, heats, and leukotriene, lipoxine. Collectively, omega-3 derived lipid mediators are known as eicosanoids. Sorry, I cannot pronounce this name. Eicosanoids is the correct name. So, generally, eicosanoids are known as inflammatory lipid mediators. Basically, you don't need to memorize those names, just get the key message. The key message is metabolites from omega-6 fatty acids are inflammatory mediators in humans, animals, and cells. Basically, they promote the inflammation. So due to inflammation, you can get these bad effects that include some increase in pain, increase in cell division, and so on. So it's been gone for decades now. So people have done several researchers and they confirm inflammatory nature of these omega-6 derived lipid mediators. Let's see the scientific evidence in animals. This is the one of the latest study. What they did was they isolate splenocytes from the rats that were fed with different arachidonic acid content. Which arachidonic acid is an example for omega-6 fatty acid. Then they measure the PGE2 content, and PGE2 is an indicator for inflammation. What they found was strong positive association with arachidonic acid percentage and PGE2 production in rat splenocyte. They found there's a linear relationship between saturated fatty acid content in plasma and omega-6 derived lipid mediators. Example is PGE2. Let's look into human study. In this human study, they isolated the human PBMC and they activated with LPS. LPS is lipopolysaccharides from microbes. Then they measured the production of omega-6 lipid mediators, namely PGE2 and LTB4. Actually, they stimulate at the 15 days after isolation, and what they found is uh, omega-3 fatty acid, omega-6 fatty acid supplementation enhance these omega-6 derived lipid mediators in young healthy men. So I want to emphasize one of the latest novel finding, and this is very fairly new, about 2 years old. So one of the several a couple of research group identify the PGE2, one of the arachidonic acid derived lipid mediator, also stop the inflammatory signal by inhibiting COX2, LOX5 enzyme. So it means it could be stopping the inflammatory reaction, but when we look into overall function of omega-6 derived lipid mediators, they are inflammative mechanistically, and we can say it is very complex. The further research are needed, but overall, omega-6 fatty acid derived lipid mediators are inflammative. Let's move into omega-6 fatty acids. There are three types of omega-3 fatty acids available: alpha linoleic acid, mainly from plant sources, and DHA and EPA. You can find only EPA and DHA from marine fish such as salmon, sardine, mackerel. And also, more recently, people are isolated these EPA and DHA from krill. Krill is a one type of shrimps, and krill oil also is available in the market right now. Out of these omega-3 fatty acid type, DHA is the most potent type. Usually, alpha linoleic acid does not show any anti-inflammatory properties, but mainly DHA and EPA show the anti-inflammatory properties. Here is the fatty acid composition of human blood cells. We see a high amount of omega-6 fatty acid. There are rather modest amount of EPA and DHA in human cells. But we know we can change this amount through the giving people more omega-3 fatty acid by eating fish or using supplement. Here is the evidence for incorporation DHA into cell membrane. DHA is efficiently incorporated into cell phospholipid when we increasing the DHA concentration. Let's look at the let's look at the DHA 25 micromolar concentration. It is a red color bar. You can see very high level of DHA level after incubation with 25 micromolar of DHA for 24 hours. Again, DHA is omega-3 fatty acid. Here is another example with animals. We fed animal with different doses of DHA and observed the incorporation of DHA into their tissues and red blood cells. What we found was DHA supplementation dose dependently increased the omega-3 Hoover index. Basically, Hoover index is an indicator for omega-3 percentages in cells. Let's see anti-inflammatory properties of DHA in cell culture studies. We incubated silica-triggered macrophages cells with DHA and then measured the production of different inflammatory markers, including IL-1 alpha and IL-1 beta. What we found was DHA dose dependently decreased the DHA dose dependently suppresses silica-induced IL-1 alpha and IL-1 beta cytokine release. Let's just look at IL-1 expression between 10 micromolar concentration and 25 micromolar concentration. You can see high level of inhibition for both IL-1 alpha and IL-1 beta and IL-1 alpha. Both IL-1 alpha and IL-1 beta is indicator for inflammation. DHA is omega-3 fatty acid. Let's see results from several clinical studies over past 30 years. So you can see these clinical studies suggest DHA and EPA intake potentially decrease disease activity. Disease activity include inflammatory response and its measurement of markers we just spoke a few minutes ago. My next big question is, does how DHA supplementation affect resolution of inflammation? Answer is SPM, also known as specialized pro-resolving mediators. When cells have external stimuli, DHA further metabolites into small lipid mediators. Those lipid mediators include resolvin, protectin, and myrcins. Collectively, all these are known as SPMs. Generally, SPMs are known as anti-inflammatory lipid mediators. Can you remember omega-6 derived lipid mediators are inflammatory, but DHA-derived lipid mediators are known as SPMs. They are anti-inflammatory. These are the pathways for anti-inflammatory mediators from omega-3, and you don't need to memorize these names just for information. Basically, main function of SPM is to block the inflammatory response in these cells, in the cells, animals, and humans. Here is the summary. So basically, when we have a trigger, it activates the inflammation, which SPMs are the metabolites from omega-3 fatty acid that help in resolution of the inflammation. This process block the autoimmunity development of autoimmunity and chronic inflammation. But when someone has high amount of omega-6 fatty acid, what happen? Lipid mediators from omega-6 fatty acids, those are inflammatory. They promote chronic inflammation, and it could lead to autoimmunity and different chronic inflammation. So this is depiction of the summary from different way. I'm trying to explain. Triggers activate the triggered is the initiation for the fire. Fire means inflammatory response. And SPMs means omega-3 fatty acid derived lipid mediators. These help to control the fire. But omega saturated fatty acid and omega-6 fatty acid main function is to maintain in the fire. So here's the summary. Saturated fatty acids and omega-6 fatty acid maintain the inflammation, which continues the autoimmunity and chronic inflammation. And omega-3 fatty acid derived lipid mediators, SPMs, play key role in resolutions, and it's also help to block the autoimmunity and chronic disease conditions. So I know there are a lot of dietitians and nutritionists in this seminar. So I thought to give recommended intakes for omega-3 fatty acid. HMA means American Heart Association says taking up to 3 grams of omega-3 fatty acid is safe. And FDA does not recommend intolerable upper intake level for the DHA, but IOM is Institute of Medicine. They have given the adequate intake for um, omega-3 fatty acid. Usually for the adults, it's something range around 1.2 to 1.6 for male and 1 to 1.1 for females. So, and I want to emphasize one more thing. So most of the fish oil label is misleading. As a dietitian, you need to prescribe the omega-3 amount. So if you see if you see here, you can see here two values. One is fish oil amount, 1000 milligram, but it has only less amount of omega-3. When you prescribe, you need to prescribe the actual omega-3 fatty acid content. Otherwise, it is not effective. If you give the less amount of omega-3, it is not effective. You cannot get the benefit of omega-3 fatty acids. So now I have already spoken first three part of my presentation. Now I'm going to move into case study. I'm going to talk about lupus. Let's give some introduction about lupus. Lupus is a devastating systemic autoimmune disease that affects approximately 1.5 million people in the USA. It is characterized by inflammation, losses of self-tolerance, which lead the development of autoantibodies. This uncontrolled inflammation then becomes systemic. As you see in this picture, lupus affects a variety of organs, including skin, central nervous system, lung, spleen, joints, so on. Prior to modern day drugs, individuals with lupus often crumble to renal failure as a result of deposition of autoantibody complexes in kidney. There's no permanent cure for lupus, And treatments option, these are the treatment option, mainly help to suppress the symptoms. Lupus is a genetic disease. However, genetic does not tell whole story. Uh, this is there's evidence with twin studies. If one identical twins has lupus, the likelihood of the other developing disease is only 30%. This indicate that exposome play a role. Exposome can be defined as the measure of all the exposures of individual in the lifetime. Element of exposome include things we eat, such as dietary fatty acid, omega-6 fatty acid, and things we inhale, such as environmental toxins, such example is crystalline silica. Let's talk a little bit about crystalline silica. Crystalline silica is respirable particle produced when rock, sand used or processed. Crystalline silica is etiologically linked to the lupus and other autoimmune diseases that include silicosis, rheumatoid arthritis, Sjögren's disease, and lupus. Sorry. Approximately 2.1 American exposed to this silica. This include construction workers, military personnel, and mine workers. Exposure to crystalline silica also associated with some non-autoimmune diseases that include silicosis. Silicosis is not autoimmune disease, it's just a chronic inflammatory disease. So here we're trying to mimic human lupus development using NCBWF1 mice. Basically, what we did was we exposed mice to silica, basically silica exposed into their lung, then create unresolved inflammation in their lung. This promote losses of tolerance and early onset of autoimmunity along with developing ectopic lymphoid structures in germinal centers. Ectopic lymphoid structure is the one of the key powerhouse that produce the different type of autoantibodies that includes IgG autoantibodies, IgM, IgT. So as a result, we see high level of autoantibodies in lung fluids and plasma and blood. So these autoantibody complexes can deposit in the kidney, ultimately leading into renal failure and death. This is the same consequences happening in the human when they have lupus. So even in the humans, when they have lupus, we can observe high amount of autoantibodies, ectopic lymphoid structures development, and kidney failures, and eventually they are dying from kidney failures. So here we're trying to explore how DHA intake influence the silica-induced autoimmunity. We investigate how DHA impact on unresolved inflammation, ectopic lymphoid structure development in lung, and autoantibody production, and glomerulonephritis, that means kidney failure. Let's move into results. So here is the sequence that I just explained. The N is the renal failure and die. So renal failure will occur if lupus is left untreated. Here we assess using PAS staining. For the kidney, we measure the glomerulosclerosis and tubular proteinosis. In this slide, G indicate glomeruli and RT indicate renal tubulars. So in these experiments, in the animals exposed to silica, the arrow are pointing here to glomerulosclerosis. You can see here, when they treated with silica, you can see high glomerulosclerosis. But when you look into the control group and DHA and silica treated group, you don't see that kind of manifestation, kidney manifestation. So I never seen this kind of inhibition. Like I observe with some drugs, but they have several toxic effects. But when we using, we were able to see the clear kidneys same as control. And another hallmark of lupus is systemic autoantibody production. Here we assess uh, how DHA affect on the autoantibody production. So basically DHA inhibits silica-induced autoantibody production. You can see there are several autoantibody groups upregulated with silica. When we treated with DHA, those are strongly upregulated. So if you need more information, you can refer these papers. In all the slides, I'm giving some reference. You can go and get more information about all studies and more information about autoantibody production. So this is the another aspect of the lupus. It's very complex. I'm not going to trying to explain you. So just remember, development of ectopic lymphoid structure is a hallmark for all the autoimmune diseases. So basically, ectopic lymphoid structures are terrestrial lymphoid structures. So they develop in the area which have the high inflammation. Those terrestrial lymphoid structures made with B cells, T cells, and dendritic cells. The function of this ectopic lymphoid structure is to produce autoantibodies and mature the autoantibodies. So interestingly, we observe these structures were completely inhibited when we feed with DHA. You can see here, this is the control group. Uh, and when we treated with silica treated group, you can see here higher amount of T cells, B cells, and follicular dendritic cells. These are the markers for development of ectopic lymphoid structures. And when we look into the DHA treated group, you can see here clear inhibition. So finally, we measure the unresolved inflammation. For that, we performed the genomic study and proteomic study, and we found silica induced most of the inflammatory-related proteins that include chemokine, cytokine, adhesion molecules, and signal transduction molecules and growth factors. And interestingly, when we feed animals with the DHA, we observe silica-induced chemokines, cytokines, all the inflammatory proteins are significantly inhibited. If you need more information, you can refer these papers, you can get it. So this is the summary. Uh, so when silica instillated mice were fed with DHA, we observe ectopic lymphoid structure development is blocked, autoantibody production is decreased, animals have minimum kidney damage and long lifespan. These findings demonstrate that productive effect of DHA omega-3 fatty acids on lupus, and we believe similar effect will find in human lupus patient. Actually, I just want to emphasize one more thing. Our clinical findings are really promising. Lupus Research Alliance, one of the key lupus research foundation, they just started, not just like a couple of months ago. No, yeah, they started the clinical study using krill oil. Krill oil is the another form of DHA, which is isolated from krills. Krills means type of shrimps. Thank you for your attention, and I would happy to answer any questions. Oh, one more thing, sorry, I forgot to tell you. So I want to I want to thank for my mentors. I'm very honored to work with my two distinguished scientists during my postgraduate studies. Thank for their mentorships and their guidance that will really help me to get good results for my graduate studies.
Thank you, Dhananjaya. That was a wonderful presentation, and I feel very honored and proud to listen to you, my fellow friend, to see him excelling at his best element. So now the seminar is open for the discussion. If anyone has any questions, you can just unmute yourselves and can ask from Dr. Dhananjay directly, or you can just type in the chat box. So we will, revert back those questions back to Dr. Dhananjay. And Ms. Paramvi and Ms. Senari, if they have any questions that they have collected from the group, can also relay them back to Dr. Dhananjay so that he can answer some of the questions. So it's question time. So if you have any questions, concerns, they are very welcome. And of course, as Dr. Dhananjaya said earlier, if you want to ask a question in Sinhala, that's also fine, and he will be able to answer. And he would be happy to answer your questions.
Dr. Dhananjaya, so
Yeah.
thank you very much for your wonderful presentation. I'm Kumari here. So actually, so what you said is that, so based on the presentation, what I understood is that, so as the high saturated fatty acid diet, high saturated diet increase the inflammatory markers. So in your
Yes.
talk, so you mentioned about the different fatty acids, the palmitic
Yes.
acid. So that is mainly present in the Western diet with the butter. So, but consider the Sri Lankan situation, you know that our staple, the fat source is the coconut fat. So it contains lauric acid, as you mentioned. So it is also acting as as a trigger. So so it increases the inflammatory marker.
Yeah.
Yes, yeah,
So yeah, so that's a really good question. So actually, there are several clinical studies done with coconut oil and other different type of fatty acids. But the most of the time, when the person is obese, then they observe the more inflammatory markers increasing. But if they are lean, they did not observe that kind of elevation of inflammatory markers. So like it's only for the human, but these are cell culture or animal studies, and
yes.
in that case they getting the monotonous diet or the specific fatty acids, so then we can definitely see the increase of those kind of markers. When considering the humans, we observe that kind of elevation only in obese people, not in the lean people.
Okay, so that is actually, so because cell culture is studied, the control study the in vitro study. So and in that cell culture study, you found this the inducing COX expression. What
Yes.
about the other the inflammatory markers like IL-6, IL-8, interleukin 6 and 8? So any yeah relationship with the lauric acids, C12 and the
Yeah.
yeah.
Actually, this specific paper, they only measure the COX-2 and TNF
Okay,
alpha, if I'm not mistaken. There are other papers, they measure the IL-6, sometimes IL-33, IL-1 beta, IL-1 alpha. Sometimes they induce those markers, but generally, I would say most of the time, those lauric acid induce the COX-2 expression.
okay, thank you. And again, so we normally, so as an inflammatory markers, we measure the CRP, the C-reactive
Yeah.
protein. So so when I see your list, I understood that the CRP was not there in that list. So is there any reason for that? Just to clarify.
So actually here I am mainly focused on autoimmune diseases. In the
Okay.
autoimmune diseases, C-reactive protein is not one of the key inflammatory marker, but it could be. But mainly cardiovascular diseases, C-reactive protein is the one of the key marker for the inflammation.
Okay, so thank you. And my last question is what I understood finally is that there is an interaction between the gene expression. So if we eat that the high saturated fat diet or the unhealthy diet, it actually acts as a trigger to which increases the inflammatory by changing the expression of the genes. So that's what
Yes.
the yeah. Okay.
Yeah.
Thank you.
It's like
Thank you.
yeah. So sorry. Go ahead.
You know, it's fine. So that's I just want to have a discussion on that. Yeah.
Yes, mainly saturated fatty acid act on the TL4 receptor, um, tolerant receptor 4. Then it go for the NF-kappa B and activate the gene activation. So that's the pathway for saturated fatty acid. But for the omega-6 fatty acid is little bit different. But omega-6 fatty acid, they are incorporate into the cell membrane. So when there's external stimuli like LPS or microbial trigger or some other oxidative stress, those omega-6 fatty acid released inside the cytoplasm. So inside the cytoplasm, there are several enzymes, COX enzyme, LOX enzyme. They degraded those omega-6 fatty acid into other lipid mediators. Then those lipid mediators activate the NF-kappa B pathway and activate the inflammatory gene. That is the root
Yes.
for saturated fatty acid and omega-6 fatty acids. Hope you the answer
Yes.
for your question.
Yeah, yeah. Thank you very much, because I also involved in that such type of cell culturing study. So then, thank you very much for refreshing and also your very great talk and joining with us.
Thank you for your great questions too.
Hello, Dr. Dhananjaya. I'm Parami.
Yes, Parami.
First of all, I would like to thank you on behalf of our Nutrition Society to you. So there's a question, sir. We know fatty fish is the best source of omega-3, but if we think about some vegetarians, can they also get omega-3 from plant sources? And is their biological value as high as fish oil sources?
Yeah, that's a really good question. So more recently, there's a group in UK, Dr. Phillips Carter. He's a pioneer in lipid research. He performed some several studies on effect of plant source omega-3 fatty acid, alpha-linoleic acid, and EPA and DHA, their effect on inflammation. But their founding was mainly, let me show you. DHA is the highly potent bioactive in a lipid that show the anti-inflammatory property, but unfortunately, alpha-linoleic acid does not show any anti-inflammatory properties. It shows a very few anti-inflammatory properties, but they are not potent to get the real effect on humans and animals.
Okay, sir. There's another question also. So, in some researchers, they have found that there's a positive effect of omega-3 on cognitive development and other than the autoimmunity, boosting autoimmunity. So, could you please tell us, is there any importance of introducing omega-3 tablets to pregnant women at their antenatal care practices? If so, how much dosage should be given to them?
So that's a good question. If you go for the IOM recommendation, these are the recommendation for pregnant women. They recommended to intake 1.4 grams. During lactation, it's 1.3 gram. When you're taking fish oil, make sure that you are taking active ingredient, not the actual fish oil amount. It should be omega-3 fatty acid content should be something around 1.4 to 1.3 grams. Did I answer your question?
Yeah. Thank you very much,
Yeah.
sir.
And also one more input I want to tell you. In the previous question you asked, is there any option for vegetarian people? So what they can do is they can take DHA supplement from algal sources. There are several supplements using algal sources. Those are not from the fish, those are from algal, marine algal.
Thank you so much, doctor. So
Yeah.
we,
Hi, Dhananjaya. I'm Renuka here.
Hi, Renuka.
Nice to see you after a long time.
Yeah.
Yeah. Can I ask something related to Sri Lankan context? As Kumari mentioned, our diet is full of saturated fatty acids, but it's a different kind of fatty acids, lauric. But at the same time, we eat a lot of carbohydrates. Now,
Yes.
as we all know that the carbs are converted to the fats if we consume excessive amount. Of course, 65% of energy is taken from carbs in the Sri Lankan diet. Now, the endogenous fat synthesis may also affect the inflammatory reactions, irrespective of the exogenous the fats. So, what is your idea about that? Such fats synthesize as a result of excessive carbohydrate consumption.
So that's related to mainly obesity. In the fat cells, when the fats are deposited, they become inflammatory in nature. So, but I have seen when they feed with the omega-3 fatty acids, their inflammatory level is decreasing. So even either they are endogenous fats or they are like dietary fat. If we feed with some omega-3 fatty acid, it can suppress the inflammatory nature of those fatty cells in obese people. There are several researchers related to obesity and uh, anti-inflammatory nature of the DHA. Did I answer your question?
Partially, yes, but I'm wondering now the type of the fats synthesized in the liver as a result of excessive carbohydrates. What type of fats are synthesized there, and how they affect, because probably we can expect a high VLDL, and then as a result of that, the high triglyceride levels, and perhaps a high NEFA level in the blood, not postprandially, maybe the entire duration. So then I understand that the effect of a DHA and the omega-3s and the omega-6, what you said, is the standard acceptable thing, but our own the fatty acid synthesis in the body, what sort of effect? If you are eating less amount of alpha-linolenic acid, DHA, EPA, but inside the body, if such a reaction is going on, what kind of influence it can have at the end? So that's my
So it's really a
concern. Yeah.
very complex process. So we
Yes.
have to think about several factors. I mean, like um, we have to also think about the hormone regulations and other things as well. It's not just only indigenous fatty
Yeah.
acid
Correct.
production. So I think I cannot give the direct answer for that question because of the really
Yeah.
it's a very complex situation. So like I will read more about that question and get
Yeah,
back to you.
I too feel that I should also read about that. Yeah,
Mm.
yeah. So anyway, good to see you, and thanks for supporting our university students and the department by giving this nice presentation. Thank you.
Thank you so
Good
much.
luck.
So we can take just one or two questions before we wind up. Anyone would like to chime in?
Good morning, Dr. Dhananjaya. I'm Senuri here.
Yes.
When someone have risk of having autoimmune diseases like rheumatoid arthritis from genetics, will they be able to prevent the risk of developing disease by getting omega-3 fatty acids continuously since they are young age?
Uh, if they are in young age, getting omega-3 fatty acid, there's going to be definite benefit. But like if they have like rheumatoid arthritis has different levels. If they reach to end level, like means if they in the process they develop some lymphoid structures in different tissues. If they reach to that stage, it's very difficult to suppress bone decay and autoantibody production using omega-3 fatty acid. But if it is in early stage, if you take omega-3 fatty acid, it's going definitely helpful for them.
Okay, thank you, sir.
It's depend on the stage of the disease.
So can I know this also? When considering a person who already
Yeah.
have autoimmune disease, how much time it will take to get effective results from fatty acid consumption by supplements, so like that.
So also that also depend on stage of the disease. But if you take fatty acid for like 2 gram per day for 16 weeks, you will get the enough anti-inflammatory response from DHA or EPA. So basically, you need to take 2 gram of EPA or DHA for 16 weeks. Then you will see enough fatty acid incorporation into the tissues. But for the like complete resolution and complete recovery of disease, it depends on the stage of the disease.
Okay, thank you, sir.
You're welcome.
And again, my last question, sir. So, Dr. Dhananjaya, regarding the taking these omega-3 supplements for a long time, what do you think? What is your personal idea on having these omega-3 fatty acids for, for a long time? Is there any risk? Or what kind of general idea do you have with that?
So there are a lot of toxicological studies have done with even
Yeah.
very high dose of EPA and DHA, but up to 3 gram it's considered as a safe. And also FDA has not given the upper tolerance intake levels. So I would say if you take below the 3 gram per day, it's safe for long term. But if you exceeding like 3 gram, then it's going to be effect on different aspects of your like it's it could have some interaction with other drugs, and also it help to thinning your blood, and different implications can be happen if you exceed the 3 gram per day level.
Okay, thank you so much, Dhananjaya.
Welcome.
Any more questions before we wind up? We'll just give a couple of seconds for anyone who would like to raise their voice or would like to type in. So in the meantime, Dhananjaya, do you have any recommendations for us in terms of how we should balance out our omega-3s and omega-6 in our diet, just in general? Things for us to remember the next time when we incorporate our omega-3s and omega-6 to our
So
diet.
like mainly you can take omega-3 fatty acids from the fish intake. So if you can increase the fish intake, it's definitely going to help you get more omega-3 fatty acid. And also like supplements, if you are taking high amount of supplement, you can get little bit uh, sorry, if you are eating a lot of fish, then you can reduce the supplement level. But if you are not getting enough fish consumption, then you can get a little bit higher amount of supplements. And also there are two types of fish oils you can find in the market: krill oil and the fish oil. Krill oil considered as little bit higher bioavailable. The reason is that in the krill oils the DHA molecule is bind into the phosphate head. So because of that, it's binding to the phosphate head, it can directly incorporate into the cell membrane.
Okay, thank you, Dhananjay. So if there are no any more questions, I think we can wind up because, as I said, it's quite late for Dr. Dhananjay from his timeline. I'm sure he's just drinking a lot of coffee and staying awake. So we will probably let him go if there are no any other questions. So this is recorded and we will have this posted on our Nutrition Society website. So anyone who's interested in getting more details with respect to the journal articles Dr. Dhananjay has spoken of earlier, you will be able to get hold of them. And Dr. Dhananjay, if it is okay, would you be willing to share your slides with us? We will have that available to our students if that is okay with you.
Yeah, I can share some of
Yeah,
the
yeah.
slides.
So I will get in touch with you.
Some
So
published papers, except that area, I will share the other information.
sure. Sure. Thank you very much for being so generous. So we will make sure that the slides and the recording will be available on our official Nutrition Society website. So anyone who's interested in can go and get hold of the slides and the recording if you want to listen to some sections in detail. So again, thank you very much, Dr. Dhananjaya, for agreeing to participate as a guest speaker for our virtual seminar series. And thank you everyone for coming today. We had a great turnout. We had about 220 participants. So that was a wonderful turnout for us. And thank you very much, Parami and Senri, for gathering questions from the audience and directing them towards Dr. Dhananjaya. And Dr. Dhananjaya, again, thank you very much, my dear friend. It was so great to see you after so many years, even though we have kept in touch through social media. I think I haven't even seen you up close for a long time. So thank you very much. It was great. Please pass my regards to Gayatri too. I haven't seen her for a long time as well. Before winding up, I think Taruka, representing the Nutrition Society as the president of the society, would like to say a couple of words before we conclude.
Thank you, madam. Good morning, everyone. I'm Taruka Padirage, as the president of the NutSoc. And on behalf of the society, I would like to thank our guest speaker, Dr. Lichchavi Dhananjaya from United States, shared his valuable knowledge with us and allocating time for us today. Dr. Kumarat Nayaka, the head of the Department of Applied Nutrition, for guiding us in every action from the beginning, and especially Dr. Sushanti Pereira, the senior treasurer of the society, being right behind us for all the good work we do as a society. And to finally all the participants here who dedicated their time to join the session. Then last but not least, to all the members of the NutSoc for their immense support in making this event a success. Thank you very much again for gracing this session with your presence. Thank you very much.
Yeah.
Okay, thank you everyone. I think we had a very productive morning. Dhananjaya, go ahead if you want to say something. Yes,
Yeah,
please.
and also I would like to thank for the organizers for inviting me to this seminar, and special thanks goes to Shanti coordinating this seminar. And thank you.
Okay, everyone, thank you very much. As I was saying, we had a very productive early morning because we started quite early today. So tonight, now I think it's time for us to let Dr. Dhananjaya go. And probably if time permits, I think he would be very keen and generous to come and talk to us maybe later on, someday, not very soon, because he has a lot of research going on with respect to omega-3 and the area that we talked about today. So maybe in a couple of months' time, if he's available, we'll bring him back to give us a very informative and a very cutting-edge, timely topic to deliver a speech on a very timely topic. So thank you, Dhananjaya. So I'm going to conclude the session now. We will have the recording and the slides available on our website. So everyone have a great Sunday and a great week. Thank you.
Thank you.